Let's talk about T cells, the life cycle of T cells. Okay, they come from the lymphoid stem cell line in the red bone marrow. Remember, white blood cells have the myeloid and the lymphoid stem cell lines. Lymphoid is only for lymphocytes. So they're made in the red bone marrow, but they move to the thymus where they get their training. So remember, this is where they go to special ops school. So what happens exactly? Well, there are hormones in the thymus, thymosins, stimulate maturing T cells to produce antigen receptors. This is what I was talking about before. Basically, you just like shuffle a deck of cards, you randomly generate receptors, and then you stick them onto T cells, all right? So once a T cell has a receptor stuck onto it, we call it immunocompetent. That means it has its receptors in place. That means that theoretically, it's capable of recognizing its bad guy, whatever that may be. You know, again, hepatitis, measles, chickenpox, strep throat, etc. And maybe even, things that don't even exist or things from Mars, okay? Again, we just randomly generated like 1 billion different receptors, and in the thymus, they were stuck into T cells. Now, each T cell has its own unique type of receptor, and that's the only thing it will ever pay any attention to, right? We say that it's immunocompetent, meaning it now knows how to recognize that particular thing, all right? So once they are ready to go, they finish their training, now we have to deploy them, all right? Just like troops. So we call them naive T cells at this point. Naive T cells mean that although they have their receptor in place, they have never actually encountered their bad guy. So they're said to be naive. So immunocompetent cells that have not yet met the antigen they were programmed to find. So we take these naive T cells and we send them all out through the body. But before we do that, before deployment, we have to make sure that they actually are able to do their job. I mean, they have the receptor inserted, but do they really know how to use it? Do they really understand what they're supposed to do? So therefore, they have to be tested. They have to be put through tests to make sure that they know what they're supposed to do, okay? So let's talk about this. We have to select them. Now, I use a little silly example here. There were these movies with Clint Eastwood called Dirty Harry movies. I don't know if you ever saw those. I know I had a girl in my class a couple of years ago who had never heard of Clint Eastwood, so I know this is pretty old now at this point. But anyway, Clint Eastwood was a cop, and he was like the most badass cop ever. But he had this famous saying where, in all the excitement, I can't remember whether I fired 5 shots or 6. Do you feel lucky, punk, or whatever? Everybody always misrepresents what he actually said, so I'm sure I didn't get it right. But at any rate, there's this one Clint Eastwood movie where he's required to do, police officers are required to do like periodic training on their weapons. And so he was like in this abandoned like warehouse yard. And you walk through there and he's got his weapon ready to go. And then they have these pop-up targets, these like big metal pop-up targets. And the target pops up. And then what happens is then, if the target pops up, the police officer has to turn and face the target, get the gun ready to shoot. But then, as it turns out, some of the pop-up targets are bad guys, but some of them are like a little girl with a lollipop. And so, what then part of the training is, first of all, when the pop-up target pops up, you, the police officer, you have to show that you can orient to the target. And then if it's a bad guy, if it's a target of a robber, a horrible person, then you shoot. Well, not a robber probably, but, a killer or something. But if it's a girl with a lollipop, then you don't shoot, okay? So that's the idea here, all right? So notice the training has really kind of 2 parts to it. You have to be able to respond to the pop-up target. But then if it's a bad guy, you have to shoot it. If it's not a bad guy, then you have to not shoot it, okay? So that's from that movie. It's called Dirty Harry. It's actually a pretty good movie. So T cells are tested in the cortex of the thymus, all right? So immunocompetent T cells have to be able to, first of all, recognize the MHC protein. That's called positive selection. I say that's like responding to the pop-up target. If the pop-up target pops up and you don't even notice it, well, That's not going to work, because that means you're not even aware that there's a bad guy out there. So the T cells are tested, first of all, to make sure that they really are aware that they can respond to the presence of a bad guy, all right, or to the pop-up target, to the fact that something is happening out there. So you have to respond. This is called positive selection. So again, in actual physiologic terms, they have to be able to recognize the MHC protein. So as they approach another cell, they have to be able to spot the MHC protein. Then they have to not react to self-antigens. This is called negative selection. I say it's like you don't shoot the girl with the lollipop. So if the MHC protein is showing a normal cell product, then the T cell has to not try to kill it, okay? That's the negative selection. So do you get it? Positive selection, negative selection. The T cell has to respond to the fact that the MHC is even there. But then, if the MHC has a normal, harmless protein, the T cell has to not try to kill it, all right? Positive selection, negative selection. And T cells have to pass both, otherwise they're not allowed to be deployed in the body. They fail the training, and they don't get to go out there, all right? Failure to pass the test results in clonal deletion. In other words, you're killed. Wouldn't that be awesome? You took like a college class, took like one of your reading classes, and if you didn't pass, they just killed you right there. Damn, that'd be harsh. But that's what happens. So some of the T cells, if they don't pass the test, you just kill them right then and there. Or another thing called anergy. So the T cells are put into an inactive state. They're still alive, but they're unresponsive. They're basically just, they don't do anything. So I say that's kind of like being sent to the basement for the rest of your life. You're still alive, but you don't get to do anything. You don't get to go out in the world and do anything. You get sent to the basement. You have to stay there forever, okay? So, and that leaves the body in a state of what we call self-tolerance. That tries to guarantee that the T cells you send out to do your fighting are actually going to fight bad guys. They're going to recognize bad guys, but they're not going to fight your own body cells. They're not going to go after your own cells because they passed the negative selection. They're not going to shoot the girl with a lollipop. They're not going to attack cells that are displaying normal proteins. Only 2% of T cells pass. Damn, you thought this class was tough, huh? Holy smoke. Only 2% of T cells pass. This is rigorous training. Your immune system is really trying to make sure that we do what we need to do and don't end up killing our own body cells. And so these are what we call the naive lymphocyte pool. They have not yet encountered their actual antigen, but they have passed both positive and negative training, so they are deployed into the body, where they'll hang out in places like lymph nodes and in the spleen. Get it? See what I'm saying? They're going to hang out in all those places and then just watch for bad guys. That's what they're going to do. Deployed throughout the body to colonize lymphatic tissues, okay? And there you see the little showing how it's all working, okay? So you can go back and look at that if you want to. And there, that's a picture of me when I was 19 years old. No, it didn't look quite that bad. No, that's someone who's been sent to the basement for the rest of his life. Doesn't look like it? That poor guy. Yeah, that's smoking his Salems or Newports, whatever. Those menthol cigarettes. Okay, so it's time for you. Get your weapon ready. We're going to do positive and negative selection. So what's going to happen is I'm going to show you potential bad guys. And if it's really a bad guy, you have to pull the trigger. If it's not a bad guy, then you don't pull the trigger. Okay? Are you ready? Here we go. You should have pulled the trigger. That's a bad guy. All right? Get it? All right? Here we go. Here comes the next one. Should have pulled the trigger. Right? Are you playing along? You should be playing along at home. Okay, here comes the next one. Ooh, look, it's gangsters. He's holding the gun gangsta style. Ooh, watch out. You got a good chance against this guy because he's probably not going to hit whatever he's shooting at. Okay, ready? Next one. Oh! Hope you didn't pull the trigger. That's the girl with the lollipop. So see, negative selection. Don't shoot the girl with the lollipop. Okay, next one. Here we go. Oh my God, it's got bombs. It's a terrorist. Got explosives. Ooh. All right, here comes the next one. Yeah, it's a little girl, but she's got a gun. You have to shoot her. You know, you can't take chances. If she's got a gun, you have to shoot her. Oh, for God's sakes, don't kill her. Okay. Ready? Yeah. Yeah, ooh. It's a girl with a gun again. That's bad. She's firing. Oh my God. Gotta kill her. All right, here we go. Oh! Don't shoot. It's a girl with a balloon. Don't hurt her. All right, ready? Here we go. That's a girl with a big ass gun. You gotta shoot her. All right, here we go. It's a zombie. You gotta kill zombies. Okay, here we go. Texas Chainsaw Massacre. Gotta kill that, all right? Okay, here we go. I'm not sure what to do here. I think maybe we would talk first and then decide what to do next. Okay, so that was T cells. Let's look at B cells. It's very, very similar. It's essentially the same thing, all right? So they also come from the lymphoid stem cell line in the red bone marrow. They mature in the bone marrow, and B cell selection happens in the bone marrow. So remember, the T cells had to go to the thymus to get trained. B cells get to stay at home, all right? T cells had to go to like boarding school. B cells get to stay home. So B cells are also tested for recognition of MHC proteins in reaction to self-antigens. Once again, the positive, negative selection. If they fail, then they're either deleted or they go into anergy, that state of basically, you know, you're alive, but you don't do anything, inactivation. Self-tolerant B cells form B cell clones. Self-tolerant means they're not going to kill your own body cells. So we clone them. And then once again, we send them out into the body so that they can colonize, you know, the lymph nodes and the spleen and stuff like that. Okay, so it's basically the same thing. All right, so cell surface markers. This is going to be relevant to talking about the different kinds of T and B cells. So cluster of differentiation, CD, is a protocol used to classify the cell surface markers on white blood cells. So, and if cluster of differentiation isn't the name of a heavy metal band, it ought to be. If you play heavy metal, talk to your bandmates. Cluster of differentiation would be an awesome name. So look over on the right. You can see CD34, CD45, CD14, CD15. So cluster of differentiation. These again are just cell surface markers. They're little proteins on the surface of a cell, but we categorize them by type. And that's what the number is referring to. These are different types. There are tons of these different markers, okay? And they help distinguish different white blood cells from one another. So they often act as receptors or ligands. Remember ligands from Bio 201. More than 350 of these markers are now known, all right? But the different kinds of T cells, we're going to differentiate by their CD markers, okay? That's why we're talking about this right now. So, specific immunity, let's do a little overview here. So B cells create antibodies that disable pathogens and mark them for destruction. Cytotoxic T cells kill foreign cells and your own body's infected cells, and then helper T cells assist in both of the processes above. So you can kind of take a look, go through that diagram. That's a pretty good one. It's showing what's going on there. The two different, remember we have the humoral immunity and the cell-mediated immunity. B cells and antibodies are the humoral immunity. Cytotoxic T cells are the cellular immunity. All right, so cellular immunity, T cells attack foreign cells and diseased host cells, and they maintain a memory of the antigen. So there are 4 kinds of T cells. Cytotoxic T cells, which we often write as T sub C or CD8, because they have cluster of differentiation marker number 8. They're the ones who are actually the killers. They carry out the attack. When they come across, remember what do they do? How do they know who to kill? They're going to inspect the MHC1 protein. So if you encountered a cytotoxic T cell, it would inspect your MHC1 protein. If you were showing a normal cell product, They would say, hey, how you doing? Nice to see you. Hope things are going well. Let's have lunch, okay? On the other hand, if your MHC1 complex was showing an abnormal cell, that would mean that you'd been infected, and the cytotoxic T cell right there on the spot just executes you. Bullet through the brain, all right? Right then and there, no questions asked, no take you downtown, no detention, no arraignment in the morning. You die right then and there, okay? Then helper T cells, which you often know as T sub H. These are the CD4. They have cluster of differentiation number 4. They promote T cell and B cell action and nonspecific defense mechanisms. So we're going to go through that in a little more detail later, but these are really, helper T cells are the very essence. They are the very core of your specific immune system. They are the ones who are going to activate. All right, so APCs are going to dock with helper T cells. They're going to display what they've eaten on their MHC2. And then the helper T cell, if it sees a normal protein, it'll just ignore it. But if it sees some kind of abnormal protein, then it will start activating other T cells. So helper T cells are crucial for getting the whole specific immune system underway. Then memory T cells, T sub M, provide immunity against future exposure. So remember, we said that white blood cells, all blood cells, are generally short-lived. They live a matter of weeks or months. Well, memory T cells are the exception. They live for years. So once you formed memory T cells, this will happen when, if you've been infected and an APC shows an abnormal protein to a helper T cell, it's going to be the helper T cell that activates the cloning of other T cells. So the helper T cell will then cause memory cells to be formed. So in other words, once the helper T cell sees we've actually been invaded by a particular pathogen, then the helper T cell is responsible for making sure that we then create memory cells so that we remember that this pathogen has been here before. And the memory T cells are so effective because if that same pathogen tries to attack you again, the memory T cells are capable of mounting an incredibly rapid and powerful defense so that you don't get sick a second time with that same thing, all right? And then, since the immune system is so powerful, we've got to make sure that it doesn't overreact, all right? That's actually one of the problems that they're having in COVID-19. A lot of people are dying not really from the virus itself, they're dying from overreactions of the immune system. So we have what are called T regulatory or suppressor cells. Both terms get used, so either T sub R or T sub S. These are the ones that basically try to help limit the immune response so that you don't end up making it worse than it would be. I mean, we need to kill the bad guys, but we've got to make sure that we don't go overboard and end up harming our own body. All right, so after processing antigens, APCs migrate to the nearest lymph node, all right? The lymphocytic highway. Remember, the lymphatic system is the lymphocytic highway. APCs migrate to the nearest lymph node. TC and TH cells patrol the lymph nodes and respond to two types of MHC proteins. T helper cells recognize, that's their job. They respond only to an MHC2 protein on an APC. Again, they're going to read the MHC2 on that APC. If they see a normal cell protein, they don't care. But if they see a pathogen, then they're going to start the activation, all right? They're going to create clones that secrete cytokines and interleukins, which again are little messengers that allow white blood cells to talk to each other. They will also, the cytokines will attract macrophages, neutrophils, natural killer cells, T cells, and B cells. So look at how crucial the T helper cell is. It's basically setting the entire rest of your immune system, including your innate immune system, into action. So its job is basically to recognize the fact that, by God, we've been invaded by bad guys. And once the T helper cell sees that, it causes the whole immune system to spring into action, all right? And it also co-stimulates T cells and B cell mitosis and maturation so that we make tons, So remember, this is the specific immune system. So the T helper cell is going to respond to a particular, it's going to be either measles or polio or, hepatitis or whatever. Well, what it's going to do then, it's going to activate all the immune cells for that, the T and the B cells for that particular antigen, okay? So if the T helper cell sees measles, then it's going to cause the activation of measles T cells and B cells. If the T helper cell sees hepatitis, it's going to activate hepatitis T cells and B cells, okay? It's the specific immune system. So T helper cells are the mobilizers. They get everybody underway, get them mobilized. Then the cytotoxic T cells, the CD8, these are going to react. So they respond only to an MHC1 protein on a diseased cell. So they're going to check the MHC1s on all the cells in your body. If they see a normal protein, they're just going to say, hey, nice to see you, how you doing? But if they see an abnormal protein, that means it's a disease cell. And so what do they do in that point? They kill it. They release perforins, granzymes, interferons, tumor necrosis factor, etc. They release all kinds of lethal things that will kill that cell, plus things that will cause other cells to respond as well. So like the interferons. Notice they're going to secrete interferons to warn nearby cells that, hey, there are infected cells in the vicinity. All of you uninfected cells, start making antiviral preparation, all right? Prepare yourselves. And those of you who are infected, kill yourself, all right? So, see, cytotoxic T cells kill, but then also activate to a certain extent. They dispatch the infected cell immediately. Again, no questions asked. These are the contract killers, the hitmen. They are ruthless. They don't do questioning or anything. If you've got an abnormal protein on your MHC1, you die, okay? So here we see, upper left-hand corner, here's a virus, and now we have an infected cell. The virus has infected that cell. Oh my gosh. So the helper T cell says, help, help. Okay, so cool, here comes the killer T cell, and what does it do? Wait, what? Didn't it kill the wrong guy? No, it didn't. It didn't. The killer T cell, remember, it responds to the MHC1, and since this cell was infected cell is now like a member of the other army. It has to die. So the killer T cell kills your body's own infected cells. The virus in this case might be tagged by antibodies, and then natural killer cells would come along and kill the virus. But do you get it? See, killer T cells are going to kill your own body cells once they've been infected. It seems weird at first, but that's their job. Your own body cells, once they're infected, have to die. And killer T cells do that for you. So this is a great diagram right here. I recommend taking some time to work through it, all right? I found this from, online. This is from one of the A&P books. This is a great diagram. Spend some time on this, would you? It'll pay you back. It will. Here's a different book. They did it a slightly different way. Spend time on this one as well. Compare and contrast the two. See how they're similar, see how they're different. One of them might show some things that the other one doesn't. It will pay you back many times over to spend a little time to work your way through those diagrams. And then once again, here's another one. Just take a look at this diagram, okay? A lot of good info here. Different books, different artists, different authors, each portraying this in its own way, but all of them are good. All of them are true. It will really be worth your while to spend some time to look at this.